Hello again. I just got done talking about stems, and now I'm going to switch over to talking about roots of plants. So I will go ahead and share my screen. All right, so talking about roots. So some primary functions of roots are that they absorb water and nutrients from the soil, and they also anchor plants to the ground. And it's kind of cool to see this giant, like a giant tree, will have roots that are actually pretty shallow to the ground. And I don't know if you guys are familiar with hiking around in forests. When you see trees that have actually fallen over, you'll see just their root system, and it's really shallow, which is really cool to see. So, um, so if you're ever hiking around and you get a chance to see that, it's pretty cool to just kind of take a look at the roots of what was actually holding that tree up if it had fallen over. All right, so we have two different root types. We have a taproot and fibrous roots. So our taproots they primarily grow long and thick. So that's kind of what like a sugar beet is or like a carrot is. They have a really predominant taproot. And these roots will reach really deep for water, and these are usually found in dicots. So the diagram over here shows our taproot on the right plant. This looks like a sugar beet. And then for our fibrous roots, these have primarily root branches, so it's really just a big network of roots, and no single root grows longer than the rest, so they're really hard to measure, so that you can really see that none are longer than a different one. So these, they really anchor the plants to the ground, kind of like a net, like I was saying. And these are usually found in your monocots, so like your grasses. So like this looks some sort of grass. I can't tell what it is. But yeah, so which would be better for erosion control? If I had to choose, I would choose the fibrous root system because that'll actually create structure and be able to hold soil together instead of just a taproot, which is just the one root kind of creates like a cavity in the soil, so it's not going to be able to anchor the soil as well as what a fibrous root would. All right. So now we have the different zones on the root tips. So this maturation zone is the area that is kind of matured and it's doing its thing. So this begins at the first root hair. So you can see the root hairs coming off right here. And then anything from this point all the way back to the plant is going to be the maturation zone. And this is where all the cells in the roots are actually differentiated and into their specialized cell types. So like these cells in here are specialized and already differentiated and will be able to do their specific functions. And then for our elongation zone, this is where newly formed cells increase in length, and this lengthens the root. So I kind of think of this like a bendy straw, like the bend in a bendy straw. So like at first it's really compressed. So those are just like your new root cells. They're just like small little circles. And then they will increase in length. So when you pull out a bendy straw, it gets longer. So that's kind of what these are doing. They're just lengthening and are being able to lengthen that root. All right. And then the newest growth is the cell division zone. So this is the zone, area closest to the root tip. And this is, like I said, the growth zone. So this has actively dividing cells in the region. And then all of these cells in here are not differentiated into what they will be doing as far as their function in the plant. So this is just where all these new cells are being created. And then after they're created and it starts growing, then they'll move on to the elongation zone, and then eventually they'll mature and be differentiated and do their specific functions. And then also found on the tip is the root cap, and this is just kind of an area where the cells are a little bit hardier, I guess you could say, and they're able to explore in soil to find new areas without damaging the newer growth underneath them. All right, so just like with the stems, we have differences with vascular bundles between the monocots and the dicots. So our monocot roots will have the vascular bundles arranged in a circle. So if you remember back from the stems, they're kind of evenly dispersed. When you look at the roots, they're actually arranged in a circle. So we have our xylem and our phloem just kind of in a circle around the root. And then also in a monocot root, we have a pith. So this is just an area made up of parenchyma cells. And then switching over to our dicots, so like beans and trees and whatnot, we have our vascular bundles arranged in this X. So this is different from their stems. Their stems are arranged in a circle in the stems, and then in the roots, they are arranged in X. So with the xylem, you can kind of remember xylem starts with an X, so those are going to be your cells that are actually within the X, and then the phloem is going to be in the spaces in between the branches of the X, if that makes sense. So that's how the arrangement of these two. All right, now I'm going to switch over to kind of a review of everything that we've been talking about for this lab. So with our monocots, we have parallel venation on the leaves, and then for dicots, we have netted venation. So if you're looking at a plant and you just want to differentiate real quick if it's a monocot or a dicot, I like to look at the leaves. So you can do that for plants out in the field, or if you just want to take a look at, say, like a plant. This is just from my office. I have this African violet plant, and if I didn't know if it was a monocot or a dicot, one quick way to tell without doing any sort of destructive sampling is to look at the leaves. And with this, I can see that it's netted venation, so I know that it's a dicot. All right. And then for a more destructive sample or observation, like I was just saying, you can look at the stems and the vascular bundles within the stems. So in monocots, they're going to have dispersed vascular bundles, so there's really no pattern to it. But in dicots, they're going to be arranged in a ring around the stem, like in the cross section. Monocots will have fibrous root systems, so this is your grasses. So if you think of like your lawn, you know that it's, you know, good for erosion control because it's growing and nothing is eroding underneath it because of all that big root system. Whereas with dicots like sugar beets and carrots, they usually have a really defined taproot, and they're not as good with erosion control. And then for the roots, like I was saying, the monocots will have that pith in the middle, and not listed on here, their vascular bundles are arranged in the circle. So they're arranged in the circle and they have a pith in the middle. Whereas with dicots, we don't have that pith, but their vascular bundles are arranged in the X fashion, with the xylem as the X, and then the phloem in the spaces in between the X. And you can see this in this diagram. This is kind of wraps everything up here. So with our monocots, with the seed, we have just the one cotyledon. This is kind of tying back into week 4 when we were looking at seeds. And then with their roots, they have their xylem and phloem arranged in a ring. And then in their stems, their xylem and phloem is just kind of evenly dispersed throughout the stem. And with monocots, they're going to have that parallel leaf venation. So like blades of grass, you can tell the venation is just in straight lines. And then I don't think we covered this, but just extra information, flowers for monocots are arranged in threes and multiples of threes. So like their petals here, we have 1, 2, 3 petals like this, and then 3 like this, and this would be a lily. And then switching over to our dicots, which is labeled eudicots, that's just a more scientific term for that. We have 2 cotyledons, and we have the vascular bundles arranged in the X and the roots. This one's kind of more of a star. But so we have the xylem being the red, and then the phloem being these blue spaces in between the arms of the X or the star, like that. And then for the vascular bundles in their stems, they're arranged in the circle. So again, we have the xylem being red, that's towards the inside, and then the phloem is on the outside of the ring. And then we have our netted venation on the leaves of dicots, which is just a really quick identification. If you're just looking at a plant and you want to know if it's a dicot or a monocot, this is a really fast way to tell. And then their flowers, again, we didn't really cover, but their flower parts are in 4s or 5s and they're multiples. And that's all I wanted to say about this. So I will end the video here. And I will see you guys later.